In this video, we will discuss about how this HFD enhance tumor platelet endothelial cells interactions in vivo. In the previous video, we have understood the results of this HFD diet, that HFD enhances highly metastatic tumor cells to metastasize to lungs. In this video, we will understand whether it will also affect the interactions between the tumor platelets endothelial cells or not. So we use the same mice models and inject the tumors at the tail of the mice. And first we observe the platelets adheres or not, or platelets adhere to the tumor cells or not. And here is the immunostaining image in the panel A, where we can see that the platelet adheres to the tumor cells. Signals are higher in the HFD conditions compared to the ND conditions, and these things are quantified in the panel B, where we can see that in the HFD conditions, the adhered platelets by total platelets ratio is higher and is statistically upregulated compared to the case where uh, control mice, adult platelets checked in the control mice. And now they wanted to check whether the interactions of these tumors to the platelets will also cause a change in the phenotypes or shape of the platelets, as change in the platelets can say its many things about its behavior. So here the scanning electron microscope images shows that in the control case the platelets has less membrane structures formations, whereas in the HFD case the platelets are aggregate together and has more membrane structures formations. That can be seen here. Here the membranes are carbohydrate structures that are formed on the membranes are less, whereas in the case of HFD conditions carbohydrate structures formed on the membranes are higher. And these things they have quantified in this percentage of the cells, where they have find out is that aggregations formations in the HFD case is higher in the HFD mice conditions compared to the control case. That can be seen clearly in the panel D, that is FPPP aggregating is higher in the HFD conditions compared to the control case. Now they wanted to check its interactions to whether tumor platelets interactions also cause the also affect its endothelial cells interactions or not. So for that they use the HUVEC cells coated wells, and then add the platelets and the tumor cells, and what they are able to find out is that the platelets from the control mice is less adhered to the uh, HUVEC cells compared to the case where platelets from the uh, HFD diet mice, and the tumors are also adheres more in that case. And these things they have quantified in the panel F, where we can see that the area covered by platelets is significantly higher in the HFD mice conditions compared to the control case. And when they check the tumors interactions using the GFP, tumors adheres to the HUVEC cells using the GFP signals. It also shows that the tumors interactions are also higher in the case where platelets are used from HFD fed mice conditions, indicating that the platelets also affect the endothelial cells. Platelets also affect the endothelial cells interactions. Next, they wanted to, since the platelets are considered as pro-metastatic towards promoting tumors, they wanted to check platelets tumor in interactions in vivo, or platelets tumor aggregations in the lungs. So what they are able to see that when they inject the tumors at the tail of the mice, within 1 minute, in both the conditions the tumor reaches to the lungs, but after some times, after 30 minutes, they are able to see that the aggregations of the tumors by the platelets is more efficient in the HFD conditions compared to the case of compared to the case of this control mice conditions. And these things they have quantified in the panel I, where we can see that platelet aggregations index is higher in the HFD mice conditions compared to the control mice conditions. Together, all these figures or this figure indicates that HFD diet will enhance the tumor platelets endothelial cells interactions in a way that will promote tumor progressions or its metastatic behaviors. If you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. There are other figures also there for about of this main manuscript paper. Or this is just, we can say that this is just the half of the figures of this main figure 4. We will discuss other figures in our subsequent videos. Thank you for your time.